guys, welcome back to Stella in Singapore. So today I'm going to talk about how the DNA is stored in the chromosome. So before that, let's revise last week lesson. So last week we talked about nucleic acid. So do you guys remember what is the monomer of the nucleic acid? Yeah, it is called nucleotide. So what are the components? Okay, have you guys recall? Okay, the first one is sugar, base, and then phosphate. So remember that I talked about two nucleic acids, that is DNA and RNA, that we were be learning. Okay, so for sugar, right? For DNA, we have deoxyribose sugar and RNA ribose sugar, and for base, ATGC for DNA and AUGC for RNA, and we have the phosphate. Okay, so here is the figure that I got it from Chem Academy. So remember that DNA is the double-stranded, means that there should be two strands. So let's see the figure. So let me draw in the red box. It is one strand. Another strand I draw in the green box, and it's another strand. So hope you guys can see there are two strands. Here is the one strand, and here is the second strand. Okay, so do you guys remember what are the bonds between bases? Yeah, hydrogen bond. Okay, just now I roughly mentioned that DNA have the ATGC. So do you guys remember how many hydrogen bonds are there between two of them? Yeah, AT is 2 hydrogen bond, GC is 3 hydrogen bond. And I also mentioned which one is stronger. Yes, obviously GC is stronger because there are 3 hydrogen bonds while this only have 2 hydrogen bonds. Okay, another thing you guys need to remember is sugar, base, phosphate. And between two of the nucleotide, they are bound by which bond? What is this called? It is called phosphodiester bond. Okay, then the phosphodiester bond is between what? Yes, 3' carbon of one nucleotide and 5' carbon of another nucleotide. Okay, so who guys remember that? Okay, moving back to here. So you guys can see here is 5', 3'. Here is 3' to 5'. So, okay. So one strand is 5' to 3' direction. So it will be like this. Correct? Okay, another strand is 3' to 5'. Means that it is like this. The B can be ATGC, either one, adenine, guanine, thymine, or cytosine, any of these. That's why I draw 2 hydrogen bond and sometimes it will be 3 hydrogen bond. Okay, anyway, so you can see that here is the 5', 3'. Here is the 3' to 5'. So this is called antiparallel. Okay, so we can say that DNA is antiparallel. Okay, Each of these strands is called template. And this will be used in this template will be used in DNA replication, which is mainly by DNA polymerase, or it can be used for transcription, which will be covered in next video, assuming I have time. Okay, I will try to make video whenever I got some free time. Okay, so for bases, right, I already mentioned they have A, T, G, C. So whenever there is A in the template strand, whenever there is an A in the template strand, when you synthesize another strand, for example, in DNA replication, they will always put T. If there is a G, they will always put C. So this is called complementary base pair. So A and T, G and C are complementary base pair. Okay. Okay, so another concept is you guys need to know hydrophobic and hydrophilic. So hydrophobic means they love water. Sorry, hydrophobic means they hate water. Hydrophilic means they love water. So remember that in the cell, in the cytoplasm, there are a lot of fluid. So it is like for those who ever love water, they will stay outside. And for those who ever hate water, they will tend to go inside. For example, there are lots of, wait, should I use, okay, I should use blue color. So there are lots of water here. And here is the one love water, so they will tend to live outside. So green represent hydrophilic. And for those who ever hate water, they will stay inside. So they are protected by those who ever love water. Whoever love water will stay outside, and for those who ever hate water will stay inside, being protected by the one who love water. So it makes sense, right? Okay, so in this case, okay, remember DNA is the one store the genetic material. So where do they store their genetic material? It is mainly in the base. So genetic material, are they important? Yes, of course, we have to protect them. We have to protect them and prevent it from being mutated. So if once they are being mutated, there will be like diseases, which we don't want. So it has to be fully protected. So how is protected? So you can see that, sorry. So you can see that the base is inside. And this sugar phosphate is being outside. So can you assume which one is hydrophobic and which one is hydrophilic? Yes, obviously, base is hydrophobic. And sugar phosphate is hydrophilic. Is hydrophilic. And for this, we call them sugar phosphate backbone. The reason we call it like this is because this sugar phosphate backbone will protect our bases from exogenous agents. So they are not being destroyed and genetic material are protected properly. Okay, so just now, whatever I say is just a strand. So you cannot keep the strand like this. The cell is very, very tiny. You cannot keep the whole strand like this without folding. So we need to fold them. So in order to fold them, we need, so DNA is just like a strand. So we need the new, sorry, we need the histone. It's like a protein, and the DNA strand will be rotating around it, something like this. So remember, histone is a protein. So there are different subtypes like H1, H2A, H2B, H3, and H4. Okay, so H2A, H2B, H3, and H4, two sets means two of H2A, two of H2B, two of H3, two of H4. All of them together, we call them, so only these four, two sets, we call them automers. So assuming this is the histone, these two sets of histone, and this is the DNA, so they will be rotating around it, and another histone will be here, and it will be, oh sorry, it will be rotating around it, and another histone will be here, and the stranded DNA will be rotating around it. So now, sorry, I should use white. So now we have histone together with DNA. Remember, this histone is only H2A, H2B, H3, and H4, two sets, and automers. And it's called automers because they are total of 8 histones. 8 represent out. Okay, so automer together with DNA, it is called, sorry, nucleosome. It is called nucleosome. However, it's just how to say, rolling around the automer, so it's not fixed. So in order to not the DNA relax from this automer, we need something to hold them here. Something to hold them. Oh my god, sorry. Something to hold them. And it is held by the H1. It's also called linker histone. Okay, so we talk about how DNA will be together with the histone to be packed in the chromosome. So how they are interacting. So DNA histone interaction. Okay, so let me draw again DNA. The phosphate. Okay, so let me go back to last week one. So do you guys remember here that I draw negative charge? Okay, so the DNA is the phosphate, sugar phosphate backbone is negatively charged because of the presence of phosphate. Reason is presence of phosphate. So this is sugar phosphate backbone is negatively charged. So negative will attract positively charged. So which one will be positively charged here? Obviously the histone. That's why the DNA can interact with the histone. So which part of the histone is positively charged? Remember I told you guys histone is a protein. Okay, so some of you guys may know that the monomer of protein will be amino acid. So this one I haven't gone through. Yeah, so for amino acid, they can be polar, nonpolar, or charged. So here we want the positive charge. So which part of the histone is positively charged? It is the N-terminal of the lysine. Because this, okay, let me write this first. So for protein, right, they have the N-terminal and C-terminal. This one I will cover in next lesson. So for N-terminal, they have the lysine amino acid, and which is positively charged. That's the reason why they can interact with the DNA, and they found the nucleosome. So there is a hierarchy of organization of DNA. So just now we talked about the DNA strand, which is double helix DNA, and we have the nucleosome, which is the DNA together with the outagma is the nucleosome. So it's not at the chromosome level yet. So how they are going to be packed? So it will be a bit hard for me to draw out. So I took a photo from Google. Okay, so just now I see the first one will be DNA double helix, which is the double-stranded DNA. So usually DNA is this structure. That's why it's called helix. And DNA together with the outamer is called nucleosome. Okay, so here is the nucleosome. And you can see that there are lots of nucleosome together, and they will make a 10. There is a U10 here. So there is a 10. And after every cis nucleosome, they make such a 10. And it is called solenoids. Okay, you can see it is more packed here. And it is called chromatin fiber. Okay, so in this state, chromatin have two kinds, heterochromatin and euchromatin. Heterochromatin means that they are tightly packed DNA double helix. And euchromatin means that they are loosely packed. And this is loosely packed. So just now I mentioned that each of this strand 3' to 5' or 5' to 3', these are called DNA template. So they will be used as a template, and the enzyme will bind to them and produce another strand, and then produce another strand by enzyme. So for enzyme to bind to the template, they have to be loose so the enzyme can go in. Yeah. So in order for DNA replication or transcription to occur, the chromatin fiber should be at the euchromatin state. So the enzyme can bind to and then the process can happen. Yeah. So moving on, you guys can see this one. And it is called loop domain. These are hurt binder, scaffold protein. And last but not least is our chromosome. So this is how the DNA is packed in our cells. Okay, so this is the end of this video, and I hope you guys get some of the information regarding to how the DNA is packed in our cells. And maybe next lesson will be DNA replication. Yeah. Okay, so hope you guys like this video, and remember to like and subscribe my channel, and I hope I can pass down my knowledge to more and more people in the future as well. Thank you so much for watching my video, and bye bye. See you again in next video.